Well, thank you, and it's a pleasure to come to speak today. I want to thank Joe. I think we should all thank Joe and Melissa Palmer for being the main force around putting this very unique symposium or summit together. I was asked to be the first speaker, and late last night I got an email from Joe. Actually, I read it this morning. And he said, I'm really looking forward to your tour de force talk that starts this whole thing out. A little pressure, but in 30 minutes there's no tour de force here, but I do speak pretty fast. What thought I'd do in my 30 minutes, and you'll see me down here looking every so often at the time because I tend to be careful to get the time right, is to talk about new antifungal development. What are the needs? And what do we have, and do we have the answers, and a little bit about our situation with the Trehalose target, the Trehalose story. So with that as a background, I thought this was a beginning to actually try to think about fungal infections in 2014, 2015. This is a presentation a couple years ago by Gordon Brown and Ted White and several other people, just showing the magnitude of the amount of fungal infections that are actually potentially seen around the world. Both that and the mortality. And you can see up here, and I put it in kind of small letters because I have freaking no idea where in the world they got those figures. The epidemiology is still not absolutely clear to me, and if we're actually knowing what we're doing and what is the magnitude of that. But it looks really good for grants, and it looks really good for trying to go out there and say these fungal infections are really big-time problems. So what I thought I'd do is give context, because what Joe asked in the beginning is just talk about, okay, what is really happening on the bedside? Because you're going to hear some very aggressive, very detailed, mechanistic type material for antifungal development. But I thought I'd give you a frame because I can't do the big picture. I can only do what I see at Duke. It's common. We take care of fragile patients. At Duke, we'll have 100 to 110 candidemias, so that's positive blood cultures, a year. We'll see 40 cases of aspergillosis, and we'll see 15 to 25 cases of cryptococcus. I know that because I've seen that. We've reviewed it. Also, what we've done is we've reviewed what our mortality and morbidity and costs are to the management of this thing. And you can see up there for candidiasis compared to similar type patients, similar type disease processes, but they have a candidemia or they have an invasive candidiasis. Our mortality with the drugs that we have today is about 25% attributable mortality. So we still will lose a quarter of our patients to candidiasis, despite the drugs that we have today. In aspergillus, actually, it's kind of interesting. I don't know. Everybody talks about all those figures of severe disease. It is severe disease. It is a problem. It's a difficulty that we have to deal with. But actually, mortality for us is about 10%. And then finally, for cryptococcosis, no matter what we do, we see exactly this, which is 20 to 30% mortality. With all the things we have today, the amphotericin Bs, the flucytosines, the fluconazole. And there up you can see the added cost. The added cost of this thing, we calculate it right down to the last cent. With computers today, you can actually do that type of stuff and pour off all the data. And at Duke, it costs $42,000 more to have a candidemia, $25,000 more to have aspergillus, and $20,000 more to have cryptococcus. So they're costly. Now, one of the things I have to emphasize a little bit is these types of infections, and they're costly, there's still significant mortality to them. They are a price that we pay for treating the severe underlying diseases that we treat today. Let me give you an example. I started on Monday on the Transplant Infectious Disease Service. Saw 3 cases of candidemia, 2 cases of aspergillus empyema, 1 case of disseminated fusariosis, and my second case of new presentation was a case of invasive mucor in the lung. That was Monday at Duke Hospital and the kinds of patients that we actually treat. Because we treat past the time that patients may have died from their underlying disease or other types of infections, trying to treat these serious, overwhelming underlying diseases. And in that group, set the fungal infections. Now, in concept, when we talk about these things, go over them, we talk about, when we treat, we talk about prevention, prophylaxis. We talk about empiric or preemptive type therapies, and what we mean by that is empiric. We look at risk analysis, and then we make decisions when we should potentially treat these patients that are febrile. And we do some preemptive type of strategies where we use biomarkers, and I'll come back to that in a moment. And finally, we do treatment strategies for management. In this day and age, as I get older and older and older, I keep having all the house staff come up to me and various things talk about guidelines. And we do have guidelines for fungal infections. You see them. The IDSA has them. The Europeans have them. Nice little guidelines, and they all refer to me to tell me about the guidelines. Well, I write some of the guidelines. And with those type of things, the guidelines sometimes just don't fit the patients. The patients are very, very tricky and very, very complex. But we do have guidelines. We have things that we can actually start with. Now, we have tools of management for invasive fungal disease. We do risk assessment as clinicians. You sitting in this audience aren't too concerned about having an invasive aspergillus infection today or tomorrow. But if you get a bone marrow transplant, or you got myelodysplastic syndrome or acute myelogenous leukemia, and you're being treated, you're going to have to worry about that. I think in the future, hopefully, although I'm a little bit cynical, that we will understand the genetics around that. Melissa Johnson with Mikkel and Nidia have done a lot of work on Canada showing polymorphisms associated with invasive fungal infections, particularly candidiasis, and others have with aspergillus. But at this stage, they're not routine. What's routine is looking at where you're coming from, what your underlying disease is, what your exposures are, and making decisions whether these patients are at high risk and how invasive we can be at making diagnosis. We talk about prophylactic strategies, and we have prophylaxis as a possible issue to prevent the infection versus treat it. If you can prevent these infections, I would highly suggest that we try to do that. Treating these infections with high burden of organisms can be a disaster. And so if we can prevent it, we can continue to treat the underlying disease. Now, where do we prevent it today? We prevent it in patients that are really high risk, patients at probably 10% risk or greater. And we got to think about that because each individual hospital and stuff like that only has so many patients of these things. And in order to get the numbers needed to treat that is even reasonable, you're going to have to use high-risk patients, AML patients that are getting induction therapy, MDS patients, myelodysplastic syndrome patients, induction therapy, bone marrow transplant patients, allogeneic bone marrow transplant patients, patients on what I call liquid AIDS or Campath, which is an anti-CD52 monoclonal antibody that can make your CD4 count 5 for a year and a half after getting it. Those are the type of patients that we put on prophylaxis, and I think an ounce of prevention is worth a pound of cure. But we all have to look at what the risks are to these infections. Because we put them on it, there is always behind that something else, which is drug resistance. The diagnostics, the cultures, the biomarkers, the radiographs, we are in love way too much as clinicians with radiographs because we can do CT scans just like that. Or we can do MRI scans, or we can do PET scans. But I haven't seen any CT scan, MRI scan, or PET scan actually physically see a fungus. They see the reactions to these things and make decisions on that, and many of our clinicians do. The biomarkers have not been exploited very, very well, in my opinion. The beta-glucan has not been exploited very well in our strategies. The galactomannan has been used very well for aspergillus in Europe, but not very well here. We tend to pour drugs on. We look at scans. We look at risk analysis, and we don't really very carefully look at our biomarkers, and I think it's a big deficiency that we have today in the United States. Finally, we have the antifungal drugs, and I'm going to finish with that. The antifungal drugs, you know them. We've made incredible progress from amphotericin B desoxycholate. I don't think I've used amphotericin B desoxycholate in 5 years now. I think we really have gone to the lipid formulations almost invariably. It's too much toxicity. These patients are way too wobbly and they're way too sick. Azole compounds, we started the imidazoles, we went to the triazoles, the fluconazoles, and itraconazoles of the world, now the extended spectrum triazoles such as posaconazole and voriconazole. And as I heard what was it, a week and a half ago, the FDA Advisory Committee approved, sorry with this, I don't know which accent it should be on, is either savuconazole or isavuconazole. So another triazole potentially will be approved probably in March. They've been very impressive. The echinocandins, I put up there all the same. I think, again, the echinocandins have been a big, big push for us in the hospital. One of the reasons for that is lack of drug interactions. And the second one is safety and the ability to get the kinetics down pretty well and the dosing down pretty well. And they have become the drugs of choice for us in invasive candidiasis. Finally, the needs, the antifungal needs. I think we're there yet. You're going to hear now in the next day and a half about different mechanisms of action. We're going to have to tackle drug resistance. You are sitting in ground zero for drug resistance in antifungals. We've reported one of the highest incidents of echinocandin drug resistance in the world, 14% of Candida glabrata. I also came on the service the other day, and at least one of my patients was a patient with Candida glabrata resistant to azoles and to echinocandins. Some of that's the massive amount of drug we use, the sick patients we have, et cetera, et cetera. But be forewarned, drug resistance, particularly in the echinocandins and particularly in the azole compounds, is here and will continue to increase. Other things I want to challenge you to is in vivo fungicidal activity. I'm tired of treating patients for weeks, months, and years. Not acceptable. We have to have drugs that kill rapidly and efficiently and remove organisms from the tissue. We've got to be careful. Organ damage, you're using eukaryotic organisms, the fungi, and we're eukaryotic organisms, and there's surely damage that does occur, and we want drugs that not only are safe to use, because these are very, very fragile patients. They're already, kidneys are not in great shape, or lungs, or brain, et cetera, et cetera. And we use a polypharmacy. I'll average up there when I go up in noontime, because I'm going to have to do a couple consults right between this noon break. These patients will be on 10, 15 different drugs. So drug interactions become extraordinarily important to us. Finally, we need breadth. You know, what am I going to do about that fusarium kid that's 8 years old upstairs that has it in his teeth right now after he had it in his lung and in his arm? Fusarium, MICs to posaconazole, voriconazole are as high as you want to go, and even amphotericin B is 2 micrograms per ml. So we need drugs and the breadth of drugs for both mold and yeast activity. I also think we're going to need drug combinations, and I think that's the wave of the future. It's been with cancer. Why not another eukaryotic organism? I think it's going to take very potent drugs with different mechanisms of action to get the fungicidal activity that I would really like to have, where we could treat a patient for a couple days rather than a couple months. Finally, pharmacokinetics and kinetics, the issue of long half-lives, the issue of penetration into various fluid levels from the central nervous system to the, heaven forbid, urinary tract are very important to us, and some of the drugs just still don't make that. So these are important needs that we need to think about. Okay, so my last 15 minutes is to introduce you to something completely different. I hopefully introduce you to the problems and needs of the clinician up on the wards, what we see, what our issues are, and what we would like to have. The rest of this is going to be devoted to trying to find and improve those needs. Now, the first thing that started a long time ago for me was a pathway. The Trehalose pathway, a sugar pathway. It's a disaccharide. It's 2 sugars put together, as you can see up there. Cryptococcus is what we've been working with, both Cryptococcus neoformans and Gattii. And it started back with Jim Kronstadt and a colleague that we were interacting, looking at transcriptional profiling at the site of infection in a rabbit model that I've used for the last 37 years. And in that thing, we had all these genes that are upregulated, downregulated, and all over the place, and trying to figure out what's important and what's not, and that's a tricky business. We looked at this gene, the Trehalose synthase, that was actually upregulated, and it was kind of interesting as I was looking at it, because one of the critical factors was, what do we do this for? We do it to try to find targets and try to understand disease processes to eventually maybe develop drugs or vaccines. And this target, the Trehalose, We didn't have it. Mammals don't have this pathway. And I go, whoa. If this is important to fungi and to a whole series of other different, but not to us, maybe we can find selective inhibitors. So here is the pathway, and hopefully I can introduce this a little bit and give you some appreciation of it. Yi will talk about the structures when we really dial down in the next talk. It's a simple pathway going through glycolysis down here. Glucose 6-phosphate comes over here with the UDP glucose. They kind of go together with the synthase here and produce Trehalose 6-phosphate. What an interesting molecule. It can feed back on the hexokinase here. It can impact on glycolysis. It then goes on to have a phosphatase here, TPS2, that knocks the phosphate off, and there's Trehalose. That Trehalose that some people would say is a sugar that can stabilize a lot of proteins, et cetera, and surely it can. But what I've been most, as you see here, impressed with is that this is a molecule that can do a lot of different things. And you're going to see the structures just in a moment, how these structures actually keep that Trehalose 6-phosphate probably inside, because it goes outside, it can do a lot of damage. And then it goes from Trehalose down with a Trehalase here to glucose again. Now this area, the Trehalase is always an interesting area. And Steve Haas in biology here has actually looked at this area somewhat, and actually if you put crypto right in a very unique growth phase, and then you hit it with a drug like validomycin, which is a Trehalase inhibitor, you can kill this organism very, very easily, very efficiently. However, I'll tell you, we've now done the experiments in the animal model where we took that organism and grew it for 3 days just to get crypto just exactly how it should be. We put it into the rabbit, and then we gave the rabbit validomycin. Unfortunately, the crypto didn't see the validomycin. It just kept right on growing. So again, what you see in vitro and what you see in vivo, the constant interaction between these things is going to be extraordinarily important. So whatever you see in the test tube, you got to go to the next level to find out if it has activity in vivo. That said, one of the important things that I knew was glucose is the primary carbon source in the central nervous system. Mike Price did this in the past. We knocked out areas in the glycolysis, and glycolysis is extremely important. Organisms can't survive if they can't use glycolysis. They can do gluconeogenesis. That's not a problem in the central nervous system like it is in the lung. But in the central nervous system, you got to be producing glycolysis. So we knew that there was a link here. Now, in the studies that Popchai did and others did, we started knocking these genes out, the TPS1, the synthase, and then the phosphatase. And you can see here just dramatic phenotypic type issues. And I just want to emphasize here's 265, which is the Gadii strain. You knock it out, and it can't produce melanin or produces melanin poorly. And actually it shows H99 neoformans is not quite the same way. So not all crypto are created equally, and that these are different type of organisms potentially. And if you do cell wall integrity things, you can see that the TPS1 knockout here is got issues around cell wall integrity. So again, you're starting to see several areas. And if I looked at the capsule production on these things, they are also significantly have problems with capsule production if you don't have the Trehalose 6-phosphate synthase. So all those classic phenotypic things that are associated with virulence and cryptococcus are heavily controlled by this kind of simple sugar pathway. Now also, it's not in all the things, because in fact, in neoformans, it's not that way. But one thing it is with both these two, with the neoformans and Gadii, is actually with thermotolerance. And you can see here the TPS1, TPS2, they just will fail to grow at 37 degrees. In fact, this is a critical virulence factor, and you'll hear more about this with the calcineurin inhibitors, and you'll hear about the RAS inhibitors and how temperature is so important to these organisms and how you can disrupt that and potentially have these organisms produce problems with growth. Now, it turns out with in vitro, if we look at the TPS1 mutant at 37 degrees, it's really fungistatic. So in the test tube, it just won't grow, but it won't die off. With TPS2, where you build up a lot of Trehalose 6-phosphate, if you remember that pathway, it becomes very toxic and it dies at high temperature. So the two have a little difference in vitro, fungicidal, fungistatic. But let me show you what happens in vivo. These things control an incredible amount of issues around both virulence factors and other pathways, from glycolysis to apoptosis, et cetera, et cetera. And if you look in a rabbit model and you take the TPS1 mutant here, and you inject it, all are injected the same, and it's a reconstituted strain, the wild type, this organism is dying significantly by day 3, and by day 7 it's completely gone. Of all the mutants I've had, this mutant has been as fungicidal, as dying as fast as any mutant that I've ever put into the animal model, the rabbit model. Now, it's important because you say, oh, that doesn't matter. I mean, in a way, that's just the high temperature. It's not just the high temperature. We did a C. elegans model here, and you can see that TPS1 is significantly attenuated over the other strains. So it's not just a temperature situation. I got burned in a grant situation because we were doing agrobacterium media insertion. So we could make a series of TS suppressors of both TPS1 and TPS2. And they said, well, this is a terrible target because you're going to have all kinds of resistance, because you can make a lot of resistance to this thing. Actually, we were making it to try to understand how the organism could get around that temperature-sensitive phenotype. The truth of the matter is these guys are sick cookies because they have other impacts besides the Trehalose, so besides the temperature. I just want to emphasize, temperature is not all the answer here. Here's our zebrafish model, and you can see here we got a series of mutants, and the one significantly that's attenuated when you inject it into zebrafish is TPS1, and that's not done at 37 degrees or 35 degrees or 30 degrees, it's done at room temperature. So there's a series of things that these things actually have impact on. Now, Yi is going to show you some really detailed structural biology. We actually know where these TPS1 and TPS2 go, and actually TPS1 actually is in endoplasmic reticulum. You can see from our control strain here, it's in ER and probably some of the cytoplasm, where TPS2 is really out onto the membrane. I don't understand it, but I'm going to tell you I think they still get together in a complex. So even though we see them visually kind of being in two different locations, we can see it in our two hybrid systems. We can see TPS1 and TPS2 interacting, and you will see in a moment how these things actually interact with structural biology. We've also seen some of the other genes that interact with this big complex type thing, including REG1 and the heat shock protein HSP104. So there is an interaction here. There is a complex here, even though physically what we see here is that they seem to be somewhat separated. There's also pathways. And I have a whole series of pathways up in here, more about these things. And so we knew that this Trehalose 6-phosphate must go to a whole series of places. We knew it impacted glycolysis. We've already worked on it with the hexokinase 2. We're just trying to find out where all these pathways are producing temperature issues, capsule issues, melanin issues. What's happened to that? I lost it. I don't know where it's at. But I will tell you, of all those signaling mutants, the one mutant that can't produce Trehalose is HOG1. So we got really a good location, at least one of the areas where there is impact on the signaling pathways. Also, we could find out that there's an impact on what happened with these organisms as far as other pathways that they may be impacting. And this is just an RNA-seq data in a rabbit where we took the TPS1 mutant and the wild type and found genes that seem to be controlled by the Trehalose pathway, by Trehalose TPS1. And you can see a series of them up here, a lot of them in nitrogen, glycolysis, virulence. And actually you see about 5 of them up here that were in the rabbits that were controlled, that we also saw in humans when we did our RNA-seq data on human infections. So that this is not just a rabbit phenomenon. This is controlling also areas within human disease. So finally, the influence of the Trehalose pathway is diffuse. I don't have the time to tell you all the impacts that we've seen, from cell wall integrity to virulence pathways like melanin and capsule and the ability to produce disease. We actually have data, Lukash is here and has data with apoptosis and its impact. Metabolism and glycolysis in the HOG pathway. Now, I'm going to finish with my last 5 minutes is, okay, Dr. Puri told me that maybe this is a pathway, it's not seen in mammalian systems, and this might be very attractive. What are you going to do about it? Well, you're going to see, and I'm going to introduce the thing, we are looking at this as a possible target. So you can do it several different ways and try to look at this, and this is one way that we've started, but we have at least 2 other ways, because now we have structural information that you're going to see in a moment, and we could do specific fragment type, binding type studies, or we can do actually computerized modeling, because we have the structure of looking for compounds. But what we did was a classic thing of looking at inhibitors. So Jenny Tender came up with both a commercial ability to actually figure out and do enzyme inhibition assays. And you can see up here a simple thing, and in my last 5 minutes, I'm sure I'll screw this up completely, and you'll come back in your head and say, what do you really say on this? But this is a trans-screening assay for detection of TPS1 inhibitors, where you take glucose 6-phosphate, UDP, put it in, and in this type of system, we're at an antibody and labeled UDP and determine whether there is inhibition of the enzyme. I'll show you that in a little bit in a moment. We can put controls in here. We've recently, with William Shattuck, it's actually here in the audience, from Richard Lee's lab, taken a library from St. Jude's, small compounds, and looked at we put the controls in here. Our control is novobiacin, because actually it does have some inhibition of TPS1, and the small compound library. Put them all together, then add the substrates of UDP, glucose, and glucose 6-phosphate, and incubate them for a while, and then stop it, and then put in an antibody and a fluorescent UDP. Now what happens with that is that this is the reaction. If you don't get this and it's blocked and there's an active inhibitor in this type of situation, you get this thing where the thing stays, the UDP that selects the label gets onto the monoclonal antibody, and I don't understand it, but there's some physics around it, that actually the fluorescence, the mini fluorescence actually increases if you're on this big structure with both antibody and the labeled UDP. But if you don't have an active inhibitor and stuff like that, then the UDP that is being produced by the reaction competes here and takes away a lot of the fluorescent stuff that's on the antibody, and so you have a lot of free UDP, and the fluorescence is down. This actually works pretty well. And this is an example of that 1838 screen. There's a series of compounds. We had 21 compounds that actually had inhibition. This was compared to the novobiacin control, and you can see a lot of them between 50 to 100% of novobiacin, but don't get too excited about that. Novobiacin was done in tenfold higher concentrations than these inhibitor libraries. Richard has looked at some of these, and I think screens a couple of the poisons out, but we do now have compounds that actually inhibit the enzyme. And in fact, we know the structures on some of those, and some of those have characteristics that look like UDP glucose, so I think they're actually real. And we're in the process of fine-tuning that down. So in summary, we have made excellent improvement in our antifungal armamentarium. There's no question. I started years ago with only amphotericin B. Today we have 3 separate classes, and we use them extensively. But as I told you, failure still abounds. And treatment complications persist, and we need better fungicidal broad-spectrum antibiotics that are safe. Trehalose is a simple pathway for us with several critical enzymes. It's centered right in the middle of a lot of signaling for a lot of other pathways. And as I said, at least in Cryptococcus, you knock that pathway out, you've got one sick cookie of an organism. It is not found in mammals, so it should be highly selective and appears to be important across other fungal species. Because what you're going to see in just a moment is actually, I'm going to switch on you a little bit, because what he's going to do, when he comes up here, is he's going to talk about Candida albicans, Trehalose 6 phosphate synthase and the phosphatase. I know with aspergillus, because I have a friend, Rob Kramer, that's doing a lot of work with aspergillus, and it now turns out in that whole complex extraordinarily important, if not essential, to fumigatus. So the hunt is on to exploit this pathway and its fungicidal targets for us in new antifungal development. We have enzyme assays for both it and for the phosphatase. We are now screening libraries, and we now have hits, and we can dial down on. We also have structures, as you can see, that we can do modeling, molecular modeling, and we can also then do fragment analysis for these libraries. We're on the way. The hunt is on. So I want to give special thanks to our collaborators at Duke University. Unfortunately, Richard Brennan's the next speaker, and Richard emailed me on Sunday and said he's in Boston. And they couldn't get out till Friday. And I want to say right ahead of time that on Monday, Yi Mao, his graduate student, found out that he was going to give the talk. So bear with him. But I'm going to tell you, he's going to give the talk, but he did the work. And so it's a privilege, actually, and I've gone over it. He's just outstanding work on that. The other thing is, these things will never happen if you don't have collaborators. Collaborators from St. Jude's Hospital. William Shadrick is in the audience here. Richard was going to give a talk. He had another commitment that he couldn't break, but they've become our chemists to actually help us in screening these compounds and manipulating them. And finally, the perfect lab, and I put it up there, and we were out eating at a barbecue restaurant. You can see that's the type of restaurant I go to. I'm a butcher's son and stuff like that, so a lot of those cleavers are things well known to me. But this is my lab, and I want to make a special emphasis and I kind of put a halo around Jenny Tenner here, because in fact Jenny is the driving force, the technical driving force, and has made major discoveries within this pathway, and without her we'd still be back in the dark ages. So with that, I want to finish. Hopefully I started this with some excitement, with some passion, with some problems, and now we can continue to dial down, not just on the Trehalose pathway, which you'll hear about in a minute, but a series of other different pathways to try to understand how we can make a better or a complementary antifungal drug or drugs. So thank you.